So today we're going to talk about some alien genetics. I'm Dominique Brooks with Pre-College Programs. So exactly what we're going to be doing today, we're going to do an overview of basic heredity. We're going to talk about traits, genes, and alleles, and some other terms that apply to those. We're going to look at some Mendelian and non-Mendelian characteristics and traits. And then we're going to talk about some Punnett Square, so what they are, how to make those, because that's going to be very important once we get to the actual activity of making our baby alien. So we're going to determine the parent traits, and then we're going to work together to determine the traits of the baby alien. And then if there's time, maybe construct a visual of that baby alien. Once you're done with the breakouts, we're going to debrief. So hearing what happened in the breakouts, go over a possible extension activity, and then final questions. All right, so first up, we're going to talk about traits, genes, and alleles. So a trait is something that we can observe about an individual. So dimples, whether or not you have them, earlobes, whether they're attached or detached, freckles, whether or not you have them, etc. Also, so next we have the gene. A gene is a unit of heredity, and it's transferred from parent to offspring, and it's held to determine some characteristic or trait of the offspring. So basically genes determine traits. And so making up the gene, we have alleles. So allele is one of two or more alternative forms of a gene that arise by mutation, and they're found on the same place on a chromosome. So we know that our chromosomes exist in pairs, and an allele for a particular gene is located at the same spot on each of those chromosomes. So next we have a genotype, and the genotype is the genetic constitution of an individual organism, or how those alleles are combined to inform the gene. Phenotype is the physical expression or characteristics of a trait. So how that trait actually plays out. Going back to alleles, so there's a heterozygous genotype. And so as we mentioned before, there's more than one type of allele. With Mendelian traits, the allele can either be dominant or recessive. And in your heterozygous genotype, you have one of each. For the homozygous genotype, you have two of the same. So you either have two dominant or two recessive. All right, so we're going to start off with Mendelian traits, because these are the ones that people commonly think of when you start talking about genetics and doing any kind of lesson around genetics. So Mendelian trait is one that is determined by the combination of two alleles of a single gene. So oftentimes the trait is either expressed or it's not expressed. So kind of an on-off switch, or one or the other. So for example, we'll go with dimples. So this is Officer Giggle McDimples from Toy Story 4. And so she clearly has dimples. And so as we mentioned before, two alleles inform the gene for dimples. So there's the dominant, which is usually represented by the capital letter, and then the recessive, which is lowercase. And so to have dimples, you either have two dominant or one dominant and one recessive. If you have two recessive, then that means you don't have dimples. And again, those alleles are located at the same place on each chromosome. All right, so now we'll get into some non-Mendelian traits. We're going to go over 4 different types. So the first one we're going to talk about are sex-linked traits, and they're controlled by genes carried on the sex chromosomes. And it's typically the X chromosome, just because it's bigger, it has more room. And so, for example, red-green color blindness, and it's a common known fact that this type of color blindness shows up more in males, and that's because they only have one allele for this particular trait. So if that allele is dominant, then it's going to, if that allele is recessive, then it's going to manifest. For women, or for females, we kind of get two chances. So if we get a recessive allele from one parent, and we still have a chance to get a dominant from another parent. And so if we have at least one dominant allele present, then we do not have red-green color blindness. Second, we have incomplete dominance. And so this happens when the heterozygous genotype, so again, one of each type of allele is present, and it yields an intermediate phenotype. So not an either or, but when both are present, you get a third option. So the example here is hair type. So if one parent gives you an allele for curly hair, another parent gives you an allele for straight hair, the combination of those two would yield wavy hair as opposed to one or the other. Another common example in botany is the flower example. And so you have one white rose, one red rose, and you get a pink rose. That looks kind of purple, but we're going to pretend that it's pink. All right, so our third example is codominance. So this is when traits have more than one phenotype with dominant inheritance. And so that means both alleles are expressed. And so one doesn't necessarily take over the other. And so a pretty straightforward example of this is human blood groups. So we know we can have A, B, AB, or O. And the only recessive allele in this case is O. So if one parent gives you an A allele and another one gives you a B allele, then you have the combination of AB, and that's where the codominance shows up. So our last example of non-Mendelian traits is a polygenic trait. And so this trait, the phenotype depends on the genotypes of multiple genes. So not just one, but in the case of eye color, it's actually 16. And so while there are 16 genes that influence eye color, there are 2 that are responsible for most of the variants. And so if you Google it, you may see a lot of stuff that just talks about 2 genes. But the point is that more than one gene informs what your eye color will be. And so that opens up the door for offspring to have different eye color from anyone else in their families. All right, so now we're going to move on to Punnett squares, which again, they're going to be very important for the activity today. And so a Punnett square enables us to calculate the probability of each phenotype in a given offspring if we know what the parents' genotypes are. So phenotype, again, is the expression of a gene, and then genotype is how those alleles are combined and what that looks like. So here's a reminder of that. And so again, for our example, we're going to go back to dimples, right? And so we have a heterozygous mom and a heterozygous dad. And so here's what that Punnett square would look like. So you would choose one parent to put across the top, one parent to put along the side, and then that little orange space in the middle are all the possibilities for the offspring. And so here we see that there's a 25% chance down here that the child would not have dimples, since having dimples is the dominant allele. And so we have, again, homozygous dominant up here, and then homozygous recessive down here, and then these 2 would be heterozygous. So I'm going to pause there to see if there are any questions about what we've gone over so far. It was a lot of vocabulary. So if there's any confusion, I know I stumbled a few times, feel free to either unmute yourself or put it in the chat. All right, so we're going to get into our breakouts, and it's baby alien activity time. So what you're going to be working on is completing these 2 tables with your facilitator. The first table would require a coin flip to determine the genotype and then the subsequent phenotype for the second parent. And then you're going to use the first parent and the second parent. The first parent is already there, to determine what the offspring is going to look like. And with that, you're going to use a spinner or some form of random number generator. So before, again, we go into the breakouts, are there any questions about the activity or anything that I've gone over so far? All right, so we are way ahead of schedule. Jay will stick with the initial time for the breakouts. And then if you need more time, facilitators, again, just pop out into the main session and we can definitely work that out for you. So without further ado, I will have you released into the breakout sessions. All right, welcome back, everyone. I've stopped sharing my screen for a second so that I can allow you all to share. So there are a few things that I want to make sure we go over. One of those things being just in general, how things went. How challenging was it? Did you feel like it was just right, too easy? You can unmute yourselves and share in no particular order.
It was challenging for me, but my group helped me through it, so I get it now.
Awesome, awesome. That's why we like breakouts.
Emily did a great job of explaining things. She did.
I knew she would.
I think it would be a fun family activity if you had sitting around, instead of a card game, you could do the activity together. I think it might be kind of fun.
Awesome. So I see a comment in the chat that it might take more than one class in junior high to accomplish, which would be just fine.
So Kristen and I were in a breakout, and we had both kind of a background in Punnett Squares, and we really, I think we both, if I can speak for both of us, we both really enjoyed it. We tried to break ours. So in that special trait one, and I mean break by, we wanted to assign a special trait that was also linked to the X chromosome, so to the feathery expression.
Oh, wow, okay.
And so we just didn't know like how that would go. So when we came to the last one, we were like, so now it's scaly because we got the L, I guess, and then the A for us was antenna. So I'm like, so now we have scaly antennas somewhere. And so it generated a lot of good conversation of like, how would this go actually? And then wanting to come back to like check the genetics of it. Like, is it possible to have traits linked that way? Was kind of where we got to.
Okay, so it sounds like there may be a part 2 to this PD coming. I like it.
I just really wanted to share my alien really quickly, if that's okay, Dominique.
Oh yeah, we were just running down the list of share out items. Just really quickly, I know when we were talking about this PD with the team, we came up with some potential alternatives for using the Google Spinner, but did anybody get to the point to where they were talking about things to use instead of Google Spinner? Like maybe something offline?
We had mentioned in our group, they have 4-sided die. You could use a 6-sided die and respin on 5 and 6, or re-roll on a 5 and a 6. Those are the 2 kind of main options that we had talked about.
Okay. So yeah, there's some built-in flexibility with this activity. And now we will take a look at any potential renderings of the baby alien that groups were able to come up with. Feel free to share your screen, facilitators, if that would help.
Yeah, I can share ours. We didn't, and then you'll see, those were just the things that I highlighted that we wanted to come back to, that we made notes of for conversations. And then we were just a little curious about the 3 options, how those all came together. But that was our picture.
Oh, that's like, it made me think of the Teletubbies.
There's our funky antennas, which we hope are co-expressed appropriately. I'm not sure though, but we were not able to get one feathery and scaly. They look like they are a nice plush fleece material.
Okay.
And the baby is the red one. The parent is the purple one. So we did this and found a picture of the parent and part two, but then we realized that they were already in the same picture.
Yeah, no, this is good. I think it gets at some of the creativity that can come with this activity. All right, Emily.
Thanks, Dominique. So I will show our baby's traits. Let me share my screen really quickly. So we had a male, both eyes were the same color, ears didn't wiggle, hairy body hair. He was short, didn't have any feathers on his wings, blue purple fire, had retractable teeth, was a carnivore, and had smooth skin. So I will stop sharing and show you my little alien that I drew. There he is. He's got purple fire, and these are his ears that don't wiggle. Those are just fun antennas that I added for the fun of it.
Okay.
And he's blowing purple fire.
So there's a cartoon on that used to come on Nickelodeon called Real Monsters. And this definitely looks like one of those monsters.
And his teeth retract. I should have not drawn a couple of them so they could retract and whatnot. But Cece or Sue-Ann, if you drew and want to hold yours up.
Oh, what?
Yeah, I don't. Can you
Nice.
see it?
I think Cece's is giving me like a where the wild things are.
Yes.
Oh, that's good.
Look at the same.
Nice.
Oh, nice. I think you can't see mine because of my background.
I think if you hold it up directly in front of you and kind of give it to the camera. Oh, there you go.
Oh yeah, there we go. Yeah, if you use your body as the background, that's definitely.
Yeah, that's it. It's like it's sort of like a version of a Teletubby. That was what I was thinking of.
I like how you were all in the same group, but you had three very different representations, and that is kind of the point. Just in terms of you could have the same traits, but they manifest kind of differently. Very creative, and I appreciate you all taking the time to do that. And it sounds like you were very engaged. We have an extension activity to try to run through really quickly. It may be a little complicated, and I recognize that, but we're always available via email. And also, I just got word that all of the materials have been posted, so there's more detail on those as well. So I will go back to sharing my screen. All right, so we're going to take a look at the Hardy-Weinberg equation. And the Hardy-Weinberg equation is used to calculate allele frequency, but also genotype frequency. So there are a few assumptions that go along with the equation, and it'll only work for Mendelian traits that only have 2 phenotypes and 2 alleles. So again, back to that 1 dominant, 1 recessive. And so we're going to look at the frequency for the recessive phenotype and the frequency for the dominant genotype, but also the individual allele frequencies. There are a few other assumptions. So basically, we are only dealing with perfect case scenarios where there are no outside forces impacting the population. The resulting equation ends up being p squared plus 2p cubed plus q squared equals 1, where p is the, P is the dominant allele and Q is the recessive allele. And again, because we're dealing with frequencies and those are the only 2 options, they have to add together to equal 1. But if we want to talk about the gene, and so that's the frequency of 2 alleles, you have to square that. And again, so that's where our equation comes from, where the P squared is going to give us the homozygous dominant genotype frequency. 2PQ is the heterozygous frequency, and then Q squared is the homozygous recessive genotype frequency. Again, we have more materials for you that will go in depth. This link is where I pulled these 2 explanations from. It's an awesome video, and it uses penguins as an example who have yellow feet and blue feet. So then to walk through that equation, I'm just going to get all of this up here. So we're going to assume we have 100 baby aliens. And so we're going to try to complete the equation with the information that 14 of the baby aliens have ears that don't wiggle, which we know is the recessive phenotype, right? So you need 2 recessive alleles. And you start with the recessive because that's the only one that we know for sure what both of the alleles are. So that rate is going to be the Q squared. And so again, we know that it's 14 over 100, which is 0.14. And so 0.14 is going to give us our Q squared. So then from the Q squared, we can calculate Q by taking the square root of Q squared, which is 0.37. So now we know Q. Again, I know I'm just going kind of quickly, just in the interest of time. So now that we know that Q is 0.37, we can use the other part that we know, the P plus Q equaling 1, to get our P value. And so 1 minus our Q, which is 1 minus 0.37, will give us the 0.63. And so now we actually have all the information we need to complete the equation, even though it might not feel like it. So here's the equation again at the top. And then here are 2 tables that we can construct to make sure we have all of our information that we need from this equation. And so as we mentioned, our P and our Q respectively. So 0.63 is our P, 0.37 would be our Q. And then in this chart, this is our genotype frequency. So if we wanted to know homozygous dominant, that's going to be the 0.4, or our dominant squared, or I'm sorry, our P squared, which is the 0.63. Our wiggly ears, which would be our 2PQ. So that's our P times our Q times 2, which is the 0.46. And then our dominant recessive genotype, or I'm sorry, our homozygous recessive genotype, which is our Q squared, which is the 0.14. So that's a ton of math really quickly. Jay mentioned some really interesting things while we were talking about this in terms of the usefulness of Hardy-Weinberg in terms of practicality. Jay, did you want to hop in and talk a little bit about that?
Sure. When you look at those numbers, you're looking at that's what your expected genotype frequency would be in a large population. So when looking at population genetics, Hardy-Weinberg is used to kind of basically Hardy-Weinberg exists in not necessarily in reality. It's in a perfect situation where there's no selection for any of these traits. And so population geneticists are looking at where there is selection, Hardy-Weinberg will not be in equilibrium, meaning that these won't add up to 1. And so they can figure out what sorts of pressures, selection pressures there are on certain traits, and then actually calculate out different measures on how these traits are evolving within a particular population. And so there's a lot of ways that it's being used in population genetics, but when you're looking at specific individuals, it's not as helpful unless you have done this and figured out, okay, well, if we're trying to figure out what the chance of a random individual in a large population to have non-wiggly ears, we know that's approximately 14%. And so that's a question you could bring back to your students around this particular activity. If they were trying to predict the different genotype frequencies of an offspring, not knowing anything about the parents.
Awesome. Thank you so much, Jay. We're slightly over time, but there's always room for questions. So you can either unmute yourself or put that in the chat. Again, all of this information is available to you online on our page. And we're always looking at our email in case you have any questions there. But again, I understand, like, I definitely rushed through that. But yeah, I ultimately just want to thank you all for attending today. I believe all 3 of you are returners, so thank you for all of the ones you've attended. This is our final one. So I would, on behalf of all the pre-college programs, like, thank you very much for coming and engaging as much as you have. And please be on the lookout for more stuff coming from pre-college programs.
And PDUs will be sent.
PDUs will definitely be sent. And if you don't receive yours,
Let us know.
email us at precollege@oregonstate.edu. All right, take care.